The female reproductive part of a flower is represented by the gynoecium or the pistil. So this is present in the innermost whorl of the flower. The plants can have either one pistil or many pistils. So they could be monocarpellary or they could be multicarpellary. If there are more than one carpel in a flower, the carpels could either be fused, called as a syncarpous condition, or they could be free, known as the apocarpous condition. The pistil has three main parts. The uppermost part of the pistil is represented by a broad portion called as the stigma. The stigma, it acts as a landing platform for the pollen grain. The stigma then narrows down into a slender, long tube called as the style, which is helpful in transport of the male gametes. The basal portion of the pistil, it is bulged into a swollen portion called as the ovary. If you take a hibiscus plant and take a transverse section of the ovary, you will see this under the microscope. So the chambers of the ovary are called as locules. The locules are separated by a tissue. This tissue is called as the placenta. In class 9 we have studied about the different types of placentation. Attached to the placenta we have structures called as ovule. This ovule is also your Megasporangium. In the androecium, we had anther. Inside the anther, we had the microsporangium. Similarly, here we have ovary. Inside the ovary, we have the megasporangium or the ovules. In this case, hibiscus, each locule has two ovules. Some plants can have only one ovule. For example, mango or wheat, paddy. In some plants there could be more than one ovule, for example papaya, watermelon. So the number of ovules can vary. If you look at the diagram of a ovule or a megasporangium, ovule has a long stalk-like structure called as the funicle, which is what is attached to the placenta. Stalk, it fuses with the body of the ovule in a junction called as the hilum. The body of the ovule is protected by two layers called as the integuments. So there is a outer integument and a inner integument. These are protective in function. Ovule is completely covered by the outer and inner integuments everywhere except at the tip where there is a small opening. So here the integuments are absent. This opening is called as the micropyle. Therefore, this end of the ovule is also called as the micropylar end. Opposite to the micropylar end, we have the basal portion of the ovary, of the ovule. This is called as the chalazal end. Now, inside the outer and inner integuments, there is a mass of cells. So, there is a tissue, it contains a mass of cells. This tissue is called as a nucellus. So this is rich in food and nutrients. Now when the flower matures, inside this nucellus tissue is where the differentiation takes place. Like in anther, we have the sporogenous tissue where gametogenesis takes place. Similarly, in the pistil, nucellus produces a structure called as the embryo sac. This embryo sac is our female gametophyte. This is the general structure of a megasporangium or a ovule. Let us now understand how does this embryo sac form and how is the megaspore produced. Inside the ovule, megasporogenesis takes place. Ovule is the megasporangium inside which megasporogenesis occurs. The process of formation of megaspores from the megaspore mother cell through the process of meiosis is called as megasporogenesis. The megaspore mother cell is diploid, whereas the megaspores will be haploid. Let us now discuss about the megasporogenesis. Megasporogenesis takes place in the nucellus. The difference between microsporogenesis and megasporogenesis is that in the sporogenous tissue in anther, each and every cell can act as a pollen mother cell and all the cells will undergo division. Whereas in megasporogenesis, only one cell can act as a megaspore mother cell. This cell is usually present towards the micropylar region. So one of the cell differentiates and becomes the megaspore mother cell. This megaspore mother cell will undergo meiosis and it produces 4 megaspores, which is present in a line. So this is called as a linear tetrad of spores. Because there are 4 spores produced, we can call it as a tetrad. There are 4 spores, out of which the 3 spores they will degenerate, after which the top 3 spores they degenerate and only 1 spore is left. So at the end of megasporogenesis you have only 1 functional megaspore, whereas in microsporogenesis all the microspores are functional. This is the process of megasporogenesis. You get 1 megaspore which is haploid, of course. It is haploid in nature. Now, this megaspore is what will produce the structure of embryo sac or female gametophyte. So let us now discuss about the development of embryo sac or female gametophyte. So unlike the development of male gametophyte, which is very simple, the microspore tetrad will separate and you get the pollen grains. Your male gametophyte is ready and it will produce 2 cells, simple. Whereas the formation of the female gametophyte, it contains many events. Therefore, this is one of the possible 5-mark questions, either the diagram or the explanation of it. So this is the nucellus and we have 1 megaspore left, which is haploid in nature. This megaspore will obtain nutrition from the nucellus and it will grow in size. So it grows in size. Then the nucleus will divide. So step number 1, it will divide by mitosis and it produces 2 nuclei. These 2 nucleus, they then migrate to opposite poles. So one nucleus goes to this pole, another comes to this pole. So nucleus will migrate to the opposite pole. Step number 2. Later, this nuclei further undergo 2 more divisions, 2 more mitotic divisions. Mitotic divisions. Now, the megaspore, it contains 8 nucleus. Point to understand here is all these divisions are what are called as free nuclear divisions. That is, the division of the nucleus is not followed by formation of cell wall. Nucleus will divide, but cell wall formation does not take place. So then one nucleus each from one of the poles will come to the center. So migration will take place. So there are 3 cells here and 2 nucleus in the center. After this, cell wall formation will take place. Now, till now, cell wall formation has not taken place. Now, each nucleus is surrounded by a cell wall. And in the center, there are 2 nuclei. They are also surrounded by a wall. This is called as the central cell, which contains the 2 polar nuclei because they have come from the poles. So they are called as polar nuclei. There are 2. And the end where there is the micropyle, so whichever side has the micropyle, towards that end we have 3 cells called as the egg apparatus. Towards the chalazal end we have 3 cells. They are called as antipodals because they are opposite to the pole. Antipodals. Now our embryo sac, which has developed from one single megaspore, is complete. This kind of development is what is called as monosporic development. The formation of the female gametophyte, it takes place from one single spore. One spore gives rise to the entire structure. So the development is called as monosporic development, at the end of which the embryo sac contains 7 cell and 8 nucleate. The antipodals are of not much importance. The egg apparatus, the 3 cells near the micropylar end, out of the 3 cells, the center cell is usually large, and this is our egg cell. That is the female gamete. The 2 neighboring cells are called as synergids. The function of the synergid is to help the pollen tube enter into the embryo sac. The synergids, they have specialized thickenings called as filiform apparatus. This will help the entry of pollen grain from the micropyle into one of the synergids, and that is the structure and discussion about the female gynoecium. Let us quickly summarize this portion. The female part is represented by a pistil. Pistil has 3 parts: stigma, style and ovary. Inside the ovary attached it to the placental tissue, we have the Megasporangium or the ovule. The ovule has a funicle hilum, outer and integuments, inside which there is a nucellus tissue. Inside the nucellus tissue, embryo sac is formed.
The development of embryo sac is monosporic. One functional megaspore undergoes mitotic divisions to produce a 7-celled 8-nucleate condition. Embryo sac is also called as the female gametophyte, which contains the egg cell, the female gamete.